This is our report for Lab 5, respiration experiments. Before we get into what exactly we did in our experiments, we need to examine some background, beginning with the circulatory system. The circulatory system moves hormones, gases, wastes, and nutrients into and out of the cells. Nutrients are picked up in the small intestines, moved to the cells, and wastes are picked up and sent to the kidneys for removal from the body. In the circulatory system, oxygen and carbon dioxide are exchanged in the lungs and pulmonary circulation. In Figure 1, on the right side of the slide, there's a schematic diagram that shows the basic functions of the circulatory system. Next is the respiratory system. The respiratory system brings oxygen to and carbon dioxide away from the blood. The respiratory system functions through capillaries that are linked through the alveoli or the air sacs in the lungs. This enlarges the surface area of the lungs, allowing fast and efficient gas exchange. The surfactant that is excreted from the lungs reduces surface tension in liquid layer of the alveoli, thus keeping the lungs from collapsing under normal breathing conditions. These two systems work together to maintain the acid-base balance in the blood. Blood normally has a pH of approximately 7.4, being slightly basic, with that number being the ideal for the conditions. The respiratory and circulatory systems, they work to keep the blood pH between 7.0 and 7.8, which, although perhaps not being the ideal, but keeping between those ranges prevents damage and maintains the function. pH in the blood is regulated by its buffer, which is the bicarbonate system, which uses carbonic acid to dissociate into bicarbonate and hydrogen ions. Bicarbonate diffuses into blood plasma, and the hydrogen ions are bound to hemoglobin in the blood, thus keeping an increase in pH from occurring in the blood. The system buffers effectively because the bicarbonate is in equilibrium with an abundant store of carbon dioxide to be released whenever necessary. During hypoventilation or short, slow breathing, carbonic acid levels increase, and during hyperventilation or fast, deep breathing, the levels decrease, thus making the blood pH more acidic or basic, respectively. Figure 2 in the bottom left-hand corner of the slide is a diagram that shows the actual chemical reactions that happen to release the hydrogen ions and the CO2. The actual objectives of our experiments are listed on this slide. The first experiment is to show correlation between respiratory and circulatory functions and show how they work together to maintain the acid-base balance within the body, specifically the blood in this case. They also serve to show that the carbon dioxide levels provide measurable proof of the bicarbonate system buffer maintaining blood pH. And the last experiment serves to examine and better understand lung capacity. The hypotheses that we formulated for these three experiments are now listed. For the first experiment, where 2 samples of lime water were prepared, we predicted that after 1 minute of bubbling, the air bubbled by, or the lime water bubbled by exhaled air will contain more carbon dioxide than the sample that was bubbled by room air. For the second experiment, which was the effects of exercise on carbon dioxide production, we predicted that the sample that was bubbled by air exhaled after 2 minutes of exercise would contain more carbon dioxide than the sample, either of the other samples. For the vital capacity experiment, we expected that our class data would be normally distributed and show that larger people will have greater vital capacities. For the materials and methods, we followed the procedures that are outlined in Part 4 of the Lab 5 procedure document, and we only deviated once, which is where we omitted the step of normalizing the data collected. In the breathing experiment, we just entered the raw data into the spreadsheet.
Moving on to results and discussion. Part A of the experiment was comparison of room air with exhaled air. 2 50 milliliter samples of lime wire concentrated with calcium hydroxide were bubbled for a minute each. one by pipetter and the other by exhalation. Figure 3, which is shown on the left, is our samples of lime water. Sample bubbled by room air is shown on the left, and the sample bubbled by exhalation is shown on the right. Lime water becomes cloudy when it comes in contact with carbon dioxide, thus showing that air that is exhaled contains more carbon dioxide than air in the lab, thus supporting our hypothesis. Part B, effective exercise on carbon dioxide production. 3 different samples of phenolphthalein were prepared. One was bubbled with room air, one was bubbled with exhaled air, and the last one was bubbled with air after 2 minutes of vigorous exercise. Iodine was used as a reacting agent to phenolphthalein to show the carbon dioxide levels. This is our personal data table for the breathing experiment. It shows room air, exhaled air, and exhaled air after 2 minutes of exercise. It took 2 drops for the solution to turn from clear to pink with room air. With exhaled air, it took 55 drops, and with exhaled air after 2 minutes of exercise, it took 93 drops. An ANOVA analysis was performed for our breathing experiment. The p-value was 1.76 times 10 to the negative 12, showing that it is not due by chance, but statistically significant. A linear regression was also performed on the breathing experiment. There was a very strong correlation of almost 1, which indicates an almost perfect correlation between room air, exhaled air, and exercise air. The results were as predicted. The levels of carbon dioxide were different under each condition. The most carbon dioxide was produced by the exercise air, followed by the resting or exhaled air, and then the room air. During fast, deep breathing, exercising, the levels of carbonic acid decrease, making the blood more basic. This results in an increase in carbon dioxide exhalation. The linear regression analysis shows that the class's data followed the same trend as our own data. There was a very strong correlation between the number of drops of iodine and the type of air, either room, exhaled, or exercise air. The p-value, as mentioned before, 1.76 times 10 to the negative 12, obtained from the ANOVA, shows that the data was not due to chance. It was statistically significant, also supporting the correlation noted in the linear regression analysis. This is based on the entire class's data, which includes our own. Part C, measurement of vital capacity. This is a histogram showing the class data for vital capacity. The majority of the students is around 4 liters for vital capacity, as shown in the histogram. Discussion of vital capacity experiment. Our vital capacities were slightly higher than the class's mean vital capacity. This was due to the fact that we are males and also above average height. However, the vital capacities were close to the predicted values for our heights on the given chart. There is a strong correlation between height and vital capacity, which would explain the slightly higher vital capacities. Our vital capacities were in the normal range and the data was normally distributed. The mean was 3.466 liters and the standard deviation was 0.7924.